Welcome to Berry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome everybody to In the Interim, a podcast on all things science of clinical trials, statistics of clinical trials, and we pose this question of in the interim and adaptive trials, and so we've got some talk of other adaptive trials today. So I have some really cool guests. Today we're going to talk about a particular paper that came out. We'll introduce the paper, and I have my co-authors here. So Dr. Kert Viele, who sometimes hosts this show and has done a number of the podcasts, and Dr. Joe Marion and Dr. Lindsay Berry have joined me today. So welcome, everybody.
Hey, Scott.
Thanks.
So what is the paper? The paper it's been published. The title is Optimal Sample Size Division in Two-Stage Seamless Designs. So as Joe just reminded me, the math isn't necessarily new here, but somewhat to me a bit inaccessible. And so what is the problem and what are we doing? Historically, Louis Scheiner referred to this as there's learning trials and there's confirming trials. You run a phase 2 trial, you look at the data, you make decisions. You run a confirmatory 2-arm phase 3 trials, and these don't mix. Well, the world is changing a little bit, and as Don Barry always says to us, you're always learning, and you're always confirming, you're always doing both. So we're going to meld these together into a seamless phase 2-3 trial design. So a very simple example of this might be 3 doses and a placebo in stage 1 of the trial. We're going to confuse and say phase 2, stage 1. So stage 1 of the trial, maybe 50 patients on each arm, fixed randomization. Pick a dose seamlessly. The next day, you're enrolling to that dose and placebo, maybe 100 patients on each of those, the selected dose and the placebo. Seamless refers to this moving from stage 1 to stage 2 of the trial. This is made by algorithm or by DSMB, but made within a couple days, and now you're just enrolling part 2. And these will be inferentially seamless in that the analysis at the end of stage 2 is going to include the patients from the first stage of the trial and in the second stage of the trial. So these trials, you'll read about them, you'll see about them. What was inaccessible to me is we're used to group sequential type trials where you spend alpha. So I'm speaking way too much, and I said I was going to be the host. So Kert, why do we have to adjust alpha at the end when we're combining stage 1 and stage 2 together?
Well, so I mean, if you're looking at multiple doses, you have multiple chances for each of those doses to be picked and go on into the next phase. When you're actually using that data, you expect that whatever dose you pick, it might have a slight upward bias, because it was picked among multiple options. And so whenever you see that kind of cherry picking, you expect that there needs to be some kind of alpha adjustment that goes with it. And so what we tried to do here was to have nice, simple formulas for here is the adjustment, here's something, you take 2 seconds on your computer and boom, this is the threshold that you need, and you'll control alpha. You'll control type 1 error.
Okay, when I've seen these, historically we've designed these trials, there's a procedure out there for controlling type 1 error. The closed testing procedure, Brett's et al. I find it awkward, somewhat inaccessible that you combine the p-value from both parts of this, and we're going to come back to this. but it doesn't do what we do in a group sequential trial where we understand you need to control type 1 error, and we could adjust alpha at the end. Can we figure out what alpha we need at the end that's lower than 0.025? We're going to talk about one-sided type 1 error in that's lower than that, but the closed testing procedure doesn't give you that. So can you do this? So this was, then it becomes very accessible, almost like group sequential. So Joe, how did we, I know this math is out there. How do you solve this problem? How do you calculate an alpha at the end of stage 2, given the trial design?
I mean, maybe we should tell the story a little bit of like how this came to be, right? Like you came to us as like a company with this question, right? We have a weekly meeting where we talk about statistical topics, cool things we're doing in designs, hard problems we don't know how to solve. And you brought this topic. You said, I want to figure out how to do this adjustment. And so I think, and this is something that like Lindsay had worked on a long time ago, right? Lindsay, when did you first start working on this?
Yeah, that's right. I worked on this a little bit in my undergrad at UT Austin. I was working on my thesis with Peter Mueller at UT, and we addressed this, and the approach we took there was using simulations to find the adjusted alpha threshold that you would use at the end of a phase 3 study, which is one approach to doing this. And at the time, I wasn't aware of the math. It exists
yeah, it
for
speaks
finding
to
this.
that point, right? That we just like didn't know this was out there, right?
Yeah.
So what is the math? How do you solve? Simulation straightforward, but how do you do the math? What can you set this up?
Yeah, so this comes from group sequential theory. And I think Kert and I both, after you gave that talk, I think Kert and I both went away and tinkered individually, and within a week we both had a solution to this, right? And the idea is that at the interim analysis, you have a z-statistic. Your data can be represented by a z-statistic that sort of is approximately normal. And you have a z-statistic in each of your treatment arms. And then you can and because you know that's approximately normal, you can get a closed expression for the CDF of the maximum. So the math assumes you choose the best dose, the one with the largest z-value. And then you know that your data that you collect in the second half also follow a z-statistic. And so you can use quadrature to figure out sort of the exact correction you need, controlling for the correlation between the data at the interim analysis and then the full data set that you have at the end. So that's kind of the math. It's nice, normal theory kinds of stuff. I think it's 3 or 4 lines worth of math. It's very elegant. It produces a very nice solution.
I think one of the
Okay. So
Scott, one of the things I thought really is nice about the intuition here is it depends on where the interim is. So if the interim is very, very early, so you do a really tiny phase 2 and a big phase 3, you're not reusing a lot of data. You don't have to take a penalty because there's virtually no reuse. On the other hand, if the interim is very, very late, you basically spend all of your time in phase 2. It's not quite a Bonferroni, but it gets pretty close to it. So the extremes, the real question is, well, where am I going to put the interim? And I think this gets into the part where the paper starts to break a little new ground, is knowing what to do with a fixed interim. Where do you put the interim to get the optimal design?
Yep. So you brought that up, the 2 extremes. Suppose we ran a seamless 2-3 trial where I gave an example of 3 doses, 50 patients on each. But if you ran all 3 doses to the end of stage 2, and you go to the end, and now you can pick any one of the doses to be statistically significant, you get an alpha that comes out of this that's essentially Bonferroni. You get 0.0833, 0.025 divided by 3, because you carry them all the way to the end. If you picked a dose and you didn't enroll any patients in stage 1 and you run a 2-arm trial, you get 0.025. So this calculates as a fraction of the stage 1 size what your alpha spend is. And we have R code for this. By the way, did we provide the R code and so people can access the R
it's in the
code as part of the
supplement.
paper?
It's about a
Yeah.
page. It's like plain text. It's not a package.
So, by the way, the paper is in Pharmaceutical Statistics. Came out, and it's Berry et al. So, optimal sample size division and two-stage seamless designs. If you want to grab that code. So, Lindsay, I know you have the R code right there in front of you. So, if I'm going to run a trial that is 3 doses, 50, 50, 50, 50 in stage 1, and then 100 on the selected dose and 100 on placebo. So at that point, you've picked a dose and now you're enrolling those two. What is my alpha at the end of that trial? If I include the 50 from the dose selected and placebo from stage 1 with the 100, so I have 150 on each arm, what's the alpha that I use at the end?
Yeah, so in that example, your information fraction is a third. So a third of the patients in your analysis are coming from phase 2. And in that example, your adjusted alpha would be 0.013, essentially. So slightly higher than the 0.0083 from a Bonferroni adjustment.
Okay, and so you can calculate. Now you can run the trial, select your dose, run the end of part 2, and you can run with this adjusted alpha at the end of it. Okay. Now, I know we have a picture of what the alpha spend as a function of the information fraction. And I was fascinated by what would that look like. Is it linear? Is it, I don't know, is it concave up? Do you spend a lot of the alpha initially? Does it take to the end of that? Who wants to describe these curves for people who cannot see them?
I nominate Lindsay.
I feel like we should just share it, right? Well, I mean,
Well, most people consume the podcast only, they're driving to work. Only about 10% of people actually see us.
that's fair. I feel like all the best podcasts are about describing visual things.
Yeah.
So this is very on point for the podcast.
Yeah, don't take your
method.
eyes off the road. I crashed because I was looking at an alpha chart. Not a good excuse.
Yeah.
Okay, let me ask this question, Lindsay, to describe this. If I have 3 doses, actually, our picture doesn't have 3.
Doesn't have a 3 on there. Yeah.
So let's pick 2 doses. Very, very simple. I have 2 doses in stage 1, and then I pick one and I go to stage 2. In that case, it's 0.025 if I just ran a 2-arm trial the whole way. At the end of the trial, it's 0.0125. Where is it halfway between that? What information fraction? Can you pick up from that? So that would be within that.
Halfway is about an information fraction of 0.2. So
I
if you spend...
I thought this was rather fascinating because the first time you see this graph, your thought is, oh, I'm spending a whole bunch of alpha with even a small exploration in phase 2. And I think if you just stare at that, your first thought is, oh no, I don't want to spend that much alpha. Maybe I should get out of phase 2 really, really quickly, which isn't the right answer. So,
Well, no,
anyway,
you're going ahead in the paper. So
I know, but it's okay.
That's the
sorry.
best one.
Yeah, I mean, I think what Scott's referencing here is when you look at the figure, you see these curves representing, for this number of experimental arms, what is your alpha adjustment? And from going from 0, you spending 0 time in phase 2 to 10% of your patients in phase 2, you have a huge drop.
Yep.
So the penalty, I'll use Scott's favorite term, penalty, is incurred with very little exploration in phase 2. And then with additional exploration, the added penalty you take is not very much larger.
Okay, so you said the word. And this gets to the question, is it a penalty is this a good thing to do or not? So you have to adjust the alpha at the end of stage 2, but I get the benefit of including the data from stage 1. But I have to adjust my alpha at the end. And a recent podcast, I lamented the use of the term penalty, that this is an adjustment of alpha in many circumstances. You improve power, so it's a bad term to talk about distributing or allocating your alpha. Is this a good thing to do, Lindsay, to adjust your alpha and include stage 1 with stage 2?
Yes, it is a good thing to do in terms of having higher power than the same design where you only analyze stage 2 data, but you get the full 0.025.
Okay, so we investigated this question and we looked at multiple potential dose response curve, where different number of doses, different dose response curves, and fair to say that in every single scenario, it is more powerful to include stage 1 data, adjust your alpha, you have higher power in that scenario. Okay, so here we are through the paper at this point where you can calculate with a very simple R package. It does quadrature integration, the alpha that you have at the end. You can write it in the protocol at that point. It's improved power. It's a good thing to do in that. So before I pose my next question, let me pause and make sure everybody keeps me honest where we are.
So I'll add a little caveat, which is that all the calculations in the paper sort of assume the endpoint is very quick. Right, that the time to the endpoint is fast relative to the accrual rate. And I think where this can break down a little bit in terms of being always useful is if the endpoint is actually quite long relative to the recruitment rate. You end up, I think we don't really like to take breaks between stage 1 and stage 2, right? Is that fair, Scott?
That's very fair. Operationally, it can be hugely problematic if you pause enrollment at any point in the trial.
Yeah, and so it's, I think you need a slightly different set of calculations if you've got one of these long-term endpoints. The math still works that we did. All of that is still fine. It all still plays out. But like the relative benefit of this thing kind of changes.
Yeah, so we've made reference to the information fraction, so the proportion of patients that are in stage 1 as opposed to the total size of the trial. If now you only have your primary endpoint on a subset of stage 1 patients, there can be a different information fraction calculated within that setting. And some patients in stage 1 might not actually contribute to that until later in the trial. So this is something we do quite a bit. We might employ simulation. We might just be able to calculate the information fraction. And then evaluating whether this is a positive thing to do and the difference in the sample size, there's complexity to many of these trials. I think that's very fair.
Yeah. I think the other way to think about it is like, you're over-enrolled in your stage 1 in some sense, right? You have 25 patients who've completed the endpoint, but maybe you have another 25 outstanding. They're not contributing any data to the analysis. You don't pay any penalty for not having their data, but you've still enrolled them, right? And so in some sense, making stage 1 bigger is sort of extra expensive in that setting.
Yeah, I recently did a project where it was a client had 2 doses in a control in stage 1. They were picking one of the 2 doses, moving to equal randomization, 1-to-1 in stage 2. And they had this aspect that the endpoint was 12 weeks. And so if they enrolled stage 1, they'd have data at 12 weeks. There was going to be over-enrolling, but with 2 doses, suppose you over-enrolled 10 patients on each of the arms. 20 of those patients count in phase 3. And so in some sense, 10 patients are quote unquote wasted within that. And of course, it depends on the length of enrollment and the time to that, which is evaluating each one of these trial designs. Now, the other interesting part of it was they actually had earlier information than 12 weeks, which is very highly correlated to 12 weeks. And we build that in with a longitudinal model to make that decision. Okay, so now getting to where Kert wanted to go. So seamless 2-3 top trial. You can calculate the alpha very straightforwardly. It's a good thing to do. You want to include stage 1 data as part of stage 2. What does that mean about the size of the phase 2 part, stage 1 of the trial, relative to doing separate trials, Kert?
Oh, I get called on. So one of the things that was interesting, so we explored a lot of different dose responses. So some dose responses where a couple doses are good and the rest of the doses are bad, dose responses that had a linear increase, different kinds of shapes. And one thing that was really interesting for the separate trials is depending on how the doses actually stack up, you want to do very different splits in how much of your total development is in phase 2 versus phase 3. You're really at risk of doing too few or too many if you don't get that dose response right, if there's a different number of doses that are good or bad. On the other hand, if you do the seamless, you've got the option where you get to keep that phase 2 data. So going a little bit longer, you're not, you do lose something by going farther in phase 2. You don't focus as much, but it's a lot less than separate trials. So it became a very robust decision that somewhere in the 30 to 40% of your total development ought to be spent in phase 2 in that setting. And it was very robust across dose responses. It really helped people out in sizing the trials without really having to think about it too much.
Oh, and in every single scenario, the size of phase 2, number of doses, dose response, the truth of the dose response, the phase 2 part is bigger if you do seamless trials.
Yeah, the way we approached this was we identified for the seamless development program and the separate development program, what is the optimal way to allocate your total number of patients between stages in terms of maximizing power. So you're right. Yeah, in the seamless designs, the way to allocate your patients was to put more in phase 2 to optimize power compared to the separate designs.
So another part of that is that by having a larger phase 2, you're more likely to pick the better arm in the setting. Okay, so...
And you get to keep the data from that arm, right? So if you're worried about like penalties, you're actually like, you're starting with your best data set rather than having to start all the way over again. So,
yeah, so easy to calculate alpha. Phase 2 is bigger. We're more likely to get the better dose, which, by the way, doesn't just affect stage 2 and power, but the eventual approval of the drug, the dose used, that this is a thing that I think everybody would want, is bigger phase 2. When you run separate trials, it's harder because you got to get out and you got to get to phase 3 because patent life is going. But here, when those patients included in phase 3, and by the way, you're losing the white space between trials, huge advantages of this. You're also getting a better dose, which is good. There's a lot of win-win-win in the seamless 2-3 trial design scenario. Okay, now returning to an initial question where all I said was, this closed testing procedure is awkward within it. We compared the group sequential alpha calculation to the closed testing procedure. And what did we find? Joe?
Actually, Lindsay?
Okay, Lindsay? Yeah.
Yeah, we did. So we added.
We, as in Lindsay
close-
did.
Yeah.
I think we looked up some of the math, but yeah,
Yeah.
Lindsay. Yeah.
Yeah, so we did. We implemented the closed testing procedure in one specific way, and I want to say that there's probably many ways you could, figure out the details of how to implement this. So this is just what we found in our implementation. The results overall were similar, but in some scenarios, the closed testing procedure had lower power than the seamless design with this group sequential math that Joe explained at the beginning. And it was particularly evident in scenarios when one or very few doses had an effect. You'd see the power of the closed testing procedure drop. And I think that's just a reflection of how the closed testing procedure works, where you have to test other hypotheses first before you can reject the null hypothesis on the selected arm. So that sort of blocks the ability to reject the null hypothesis and lowers your power in this scenario where only one dose actually works.
So it was a bit conservative in type 1 error in some of these scenarios. It had lower power in there. So also it does very well in terms of power and better than some of the general closed testing procedure aspects of it.
Yeah, there's a weird thing about the closed testing procedure too, where, what is your estimand or your estimate, right? I guess it's probably the mean from the pooled stages, but that's not actually where you got your p-value from. I assume there's no assume it's, you're unlikely to have a weird case where your pooled analysis doesn't, meet significance, but it's just kind of weird to me to have your test and your model kind of divorced from each other in this way. Yeah.
So that brings out in number of designs we do, people working on phase 2, 3 seamless trials, a good number of them fit into this bucket. We put things and we made it very simple. Fixed sample size stage 1, fixed sample size stage 2, select a dose and move forward. A number of these designs end up with innovations to them. Adaptive things happening in the stage 1 part. You could potentially drop some doses sooner. You could potentially do RAR on those doses. You could have adaptive sample size of stage 2 part. And then in the that was the stage 1 part. I knew I would mess up phase 2 and stage 1. So stage 1 can have various adaptations. Stage 2 can have adaptations, mostly probably in sample size. Maybe you could stop enrolling sooner. The effect is larger. Flexible sample size within those parts. So that's where this gets kind of neat to use this and add adaptations to both parts of this and within seamless trials.
So there's a couple parts to that. One question is, well, if you're going to be complicated, why worry about this result? You're going to have to simulate everything. But I do think we have a lot of clients who come to us, and one of their questions is, should I even pursue a seamless trial? And the ability to go, hey, here's the basics, here's a simple one, and here's what you gain. It's worth making that kind of broad discussion over whether it's worth investigating things further. Usually the answer is yes, it's worth investigating things further. So, but that's certainly, there's value on both sides of that, even if you move away from where the result's directly applicable.
Yeah, I think there are also a lot of interesting situations too where the result is conservative, and so you can still apply it even though you're not really following the underlying procedure that's described in the paper. So Scott was talking about having a longitudinal model earlier, right? To use your early week data, your week 12 to predict your week 52, for example. And you can apply this model in that situation. You can apply this correction in that situation. what you do is you treat the information, you calculate the information fraction assuming you had all the data at the time of the interim. And that'll give you a penalty that's kind of too big, but it's conservative. It gives you analytic control type 1 error. You don't have to simulate if you don't want to. And honestly, I found that that works really well and is like absolutely acceptable by regulators. It also applies to like the Bayesian models we do, right? You may have a dose selection criteria coming from sort of a complicated Bayesian model. You may not be choosing the best dose. You may be choosing the ED90. You can still apply this procedure in those settings, and it will be conservative, and that's really helpful. I'm always happy when I can move past like simulation-based control of type 1 error, right, and into and have that part of the puzzle solved, so I can focus on the other pieces of the design.
So in the scenarios, you might want to pick the dose that is optimal over tolerability and safety at the same time. But the selection of any dose would be controlled under this procedure. It would only be cases such as we're going to take the middle dose as long as the high dose unless it's unsafe. And if based on safety it's unsafe, we're going to take the lower dose. That's a case where you probably don't even have to split alpha in a circumstances like that. It's not efficacy that's selecting the dose, but largely in the other circumstances, we've been able to employ that. So not every scenario should do a seamless 2-3 trial, though the math is great and you should almost have to figure out why you're not doing it. But what are some areas where it might not be the right thing for them to do seamless 2-3 trials?
You have to use different endpoints in phase 2 and phase 3. We don't like it, but it would break this.
So one scenario might be that it's a relatively new disease, not a rich history of knowing even what the phase 3 endpoint would be. Sometimes the FDA says, bring us the phase 2 and we'll figure out the endpoint. It would be hard to have a seamless trial in that setting. Not impossible, but that might be a hard setting where the regulatory pathway of phase 3 is so unclear that you kind of need phase 2 to do that, is one scenario.
Yeah, I think in general, to do a seamless 2-3 trial, you need to, before the trial, when you're designing it, be able to commit to a method for selecting a dose, so an endpoint and which dose you want to select. And sometimes it might not be, you might not know how to do that yet. And you might want to see all of the data and look at all of the endpoints and kind of play around in the data and pick the endpoint yourself. So I think that might be a case where being able to look at the phase 2 data before you design your phase 3 trial might be preferable.
Yeah,
but I think it's always good
cases where I might try to push back and say, okay, you're losing 200 patients, and think hard about the decisions. But there are some circumstances where it's really hard. Another circumstances may involve just CMC issues, that the method for creating the drug is not phase 3 ready. And the FDA wants you to be using the drug that's phase 3 ready. The manufacturing is set up in a way. And so that might be a case where you can't use the phase 2 data because the manufacturing isn't even part of it. You can do operationally seamless, potentially. But that's a scenario where I've seen that's a bit of a hurdle. The other one I'll spend a little bit of time on. Go ahead, Joe.
Scott, is there a situation where you have approval to study the drug over, say, 26 weeks of exposure, something like that, but the FDA hasn't approved you to look at 52 weeks or 104 weeks, what your actual primary endpoint is, and so you really can't even collect the data you need in the phase 2?
Yeah, and it might be you don't have the data enough to give a year of exposure, but you can do 12 weeks of exposure.
Yeah,
but
you need kind of an intermediate step.
Yeah. Or you don't want to put in the resources to do a 12-month study before you get proof of concept in sort of a phase 2 trial, and you're only going to get 12 weeks exposure. Might be a scenario in that. By the way, all of these trials, seamless 2-3 trials, that we do. You put a proof of concept. You need to jump a particular hurdle of reasonable efficacy, or you don't even enact the stage 2 part. So you can build in proof of concept as part of this. And that brings me to the other one, that we do a fair amount of work with smaller biotechs, and funding becomes a big issue. And the notion is that we don't have the money to run the phase 3 part. We need the phase 2 data to raise the money for phase 3. We need that phase 2 data for that. And so we need to make that data public and go to a conference and say, look how good the data are in that setting. And if it's part of a phase 3 trial and seamless phase 2/3, that's generally frowned upon, that you unblind data as part of that. So that could be a potential restriction. It's
And
something
Scott,
we've worked a lot
that would apply to NIH funding as well, where you need the phase 2 data to get the grant for phase 3.
Yep. Now, we do a lot of work with biotechs that we try to set up objective criteria in phase 2, that it won't go to phase 3 unless a certain effect size is seen, a probability of clinical significant effect size is seen. And you could potentially set up with a funder that if that comes back positive and all you get back is, did we achieve the proof of concept threshold, yes or no? And that's done by an independent stat group. And they provide that, the funder says, yes, if you can hit that, we'll help fund it. The benefits are smaller sample size, shorter timeline. It's good for the funders in that circumstance, and just for the whole reasons that this is a better approach. So we've done a good bit of that, but that could be a constraint in a number of areas where largely regulators think that part 1 is really part of a phase 3 trial because the patients are. So that could be a potential constraint in all of this.
Yeah. Those proof of concept thresholds are really important, but they're tricky because they kind of change the way the trial looks in a lot of ways. If there's a pretty high hurdle you have to jump from stage 1 to stage 2, that's going to reduce, the thing you normally would describe as like the power of the design. Because, to win the design, it's not just meeting a significant threshold at the end of it, but it's also jumping over that hurdle in the middle of it. And I think this is like always a part of clinical trial design. It's like part of every drug development program, but it's kind of hidden if you don't do a seamless design because no one specifies really what that hurdle is. You don't ever see people are always calculating power for their phase 3 after they've hit the hurdle. And so it's kind of tough to compare these. This is a really important part of doing these designs, but it can lead to some comparisons that people find confusing, I think.
Yeah, some of them are designed in a way that there is a pretty high hurdle, but they're sort of built like, but if we hit that hurdle, we want full acceleration, we want full go. If we don't hit that big hurdle, we're happy to just have this be a phase 2 trial. And we'll analyze the data, and we might go run a phase 3 program. And others are set up where it's a relatively low hurdle. They're going to go to phase 3 most of the time and set it up almost like a futility rule. So we've been involved the whole gamut of that, which can be customized to the particular scenario the person's in.
Yeah, the high hurdle I think of is like a design that's opportunity seeking. It's like designed to go really fast if things look well, and otherwise it just turns into your normal phase 2.
Yep.
But you got to get in that mindset, and you got to convince other people that that's the right mindset to have.
Yep. All right. Any other thoughts on this paper? It was one of my favorite papers in terms of setting this up, making this straightforward, but the optimality part of it I thought was just absolutely fascinating. And the ramifications to drug development also absolutely fascinating.
Yeah, this is one of the ones we use a lot. Like I use this regularly in our projects, so that's been cool.
All right. There's R code. Joe's really nice R code in the appendix, so super easy to use. Just plug it in, and then you can calculate it in seconds. or you can just do what Scott does.
Probably
and
like
you can ask,
microseconds, right?
Yeah.
Seconds would be really long.
I was going to say, or you can do what Scott does and you can message me and Joe and we'll calculate it for you. So that's also an option.
Savage.
And they have passed me the code, and I have actually learned to run it. So if I can do it, all of you can do it.
Scott's just waiting for the Fortran version of it, right? Like that's what we need.
It will be faster, but this code is so fast that you wouldn't notice within them, I'm sure.
What do you think seamless means? Like, I think of shirts. Is that the seam? Like, it's, you can't tell where the sleeve connects to the body, or is it something else?
I think you can't tell where the stage 1 and the stage 2 part, there's no seam between them. There's no white space, which is a killer in drug development. So maybe we could call these white space free designs.
I like it.
Trademark that.
We should talk about blinding, because that's something that comes up too. Like I've had people claim that you can't do these designs because the decision to go from, I've had people claim that the sponsor isn't allowed to know when the design enters stage 2, which is not true. I think it's crazy. Is that the consensus?
We certainly have
I
run
think
them.
you're right.
So we've run them for at least a decade, right, Scott?
Yeah. Yeah. And there's actually a fair amount of information. I think what gets everybody nervous is if you were to unblind any individual patient and John Doe is on placebo or not, that this can be problematic. I mean, they still have outcomes in the trial. Or if you know exactly the effect size, there's concerns about operational bias. And the FDA guidance on adaptive trials spends about half of its time on operational bias. Does it change the patients enrolled? Do the clinical sites differ in the different parts of the trial? But you can get a certain information. You hit proof of concept. That the effect was above that. And what dose is selected even in part 2 in that. And it might spawn another phase 3 trial. So this might be the first of your 2 adequate and well-controlled, and now you need to know the dose for the other phase 3 trial. Even sizing the other trial. So there's a fair amount that gets known, but there is a line where you don't want to cross in terms of what you learn at that particular time.
And there's an argument there to get a little wonky in terms of the keeping the ratio between the arms in phase 2 and phase 3 equal. So even if you get to the end, even if there are slight differences, the control and treatment, there are no systematic differences between arms, which is important for interpretability.
Yeah, and from a patient perspective, that means you might go from randomizing like 3 to treatment to 1 to placebo to 1 to 1 in the last part, and that's something to think about.
We've done some where it's 1 to 1 to 1 in the first part, and then it's 2 to 1. So you maintain the placebo proportion throughout, and the formulas in this paper will allow you to do that.
And then the other thing I'm curious, what if you don't want to really pre-specify the dose selection criteria at the interim, but you want to let like a really small unblinded group within the company see like, I don't know, a couple of efficacy endpoints summarized by arm and maybe the safety data and make that decision kind of manual mode. Is that an option? Have we tried that?
I mean, I'm gonna think out loud here. Shouldn't that be allowed from at least the math? I mean, we're picking the best arm, so picking anything other than the best arm.
Yeah.
So the math allows
So
it.
let's separate 2 scenarios. There's scenarios where a DSMB picks the dose, and the trial sets up that we're going to allow them to pick it. Then there's no concern about unblinding, and the math all works. You can pick any dose, and the math works. I think the scenario is, does somebody at the company know something? And there are no written rules about this. That's a bit of the challenge. There are scenarios where somebody very high up in the company, a CFO, a CMO, who doesn't talk to the sites, is not involved in any way, executive level that's involved in that. And I have seen that. So there are ways there can be some level of unblinding, to the company and all of that, but you want to really keep it separate from sites, involvement with anybody who's talking to patients, to really make sure that this is, it's hard to recover if somebody says, do you have operational bias? You can't show you didn't.
So you're still double blinded, but maybe not triple or quadruple or quintuple blinded.
Yeah, and the math in that situation works out for the type 1 error, because it's conservative, I guess, but you can't really simulate that decision for power. You can't say, here's how accurate our dose selection is if you don't have the algorithm pre-specified. So that maybe is a little more challenging.
That's a good point.
All right. So thank you all for joining here in this strange place called In the Interim. For now, thank you all for joining.
Thanks.
Thanks, Scott.
Thanks for having us on, Scott.